A very good day to everyone. I'm LISHANTINI, a PhD student from Universiti Kebangsaan, Malaysia. Today, it's my great pleasure to share a part of my research work entitled Clinacanthus mutans induces apoptosis in triple-negative breast cancer cells MDA-MB 231 by enhancing M1 macrophage polarization in co-culture setting. As we are all well aware of, unfortunately, breast cancer is still a major cause of death and disability in women. Approximately 1 in 8 women worldwide will be diagnosed with breast cancer, and it is a major public health concern, particularly the subclass referred to as triple-negative breast cancer or TNBC. TNBC is classified by the absence of three main receptors that are commonly found in breast cancer, such as estrogen, progesterone, and also human epidermal growth factor receptor 2. It is also characterized as highly heterogeneous with high metastatic and invasive nature and also poor prognosis. Due to the lack of all three important receptors, patients do not benefit from hormonal-based therapy. Thus, the only treatment option is chemotherapy, which could result in many side effects. However, high infiltrating lymphocytes suggest that triple-negative breast cancer may be more immunogenic, thus making more focused research on immunotherapy. One such potential and well-characterized tumor-infiltrating immune cells that we will explore is tumor-associated macrophages. Tumor-associated macrophages represent the largest population of infiltrating inflammatory cells in malignant tumors, altering the tumor microenvironment to contribute to the regulatory process of tumor progression. Tumor-associated macrophages are primarily differentiated into two distinct phenotypes, which are classically activated M1 phenotype and also the alternatively activated M2 phenotype. M1 phenotype are associated to its pro-inflammatory and anti-tumoral properties by regulating the inflammatory cytokines, which leads to tumor suppression, whereas the M2 macrophages shows anti-inflammatory and pro-tumoral effects, which supports tumor immunosurveillance, escape, angiogenesis, and matrix remodeling, which contributes to tumor progressions. Therefore, the idea of exploring the macrophage plasticity through reprogramming tumor-associated phenotype into a M1 active phenotype is emerging as a potential therapeutic approach for cancer management. Immunomodulation by plant-derived products are of active interest in therapies being designed to combat cancer. Clinacanthus mutans is locally known as belalai gajah, or in English it is called Sabah snake grass. It is one of the important species from Acanthaceae family, which has been used as vital herbal medicines in tropical Asia. It has gained much attention due to its immunomodulatory and anti-cancer effects. However, its underlying mechanisms in tumor microenvironment remains unclear. Given the key roles macrophage plays in tumor progression, we aim to study whether Clinacanthus mutans can exert its anti-tumor effect through immunomodulatory effects on polarization of macrophages. This will be achieved by two main specific objectives, which is to investigate the effect of Clinacanthus mutans on the polarization of THP1-induced macrophages and to examine the effect of Clinacanthus mutans-induced macrophage polarization on the cytokine expression level and also mode of cell death of TNBC cells. To mimic the tumor microenvironment, we first established an in vitro co-culture model of THP1-induced macrophages with MDA-MB231 cell line, which is a triple-negative breast cancer cell, to illuminate the important role of macrophages in triple-negative breast cancer microenvironment. The cells were then treated with 100 microgram per ml of Clinacanthus mutans for 48 hours. First, to investigate the effect of Clinacanthus mutans on macrophage polarization, macrophages were analyzed for their cell surface marker HLA-DR and also toll-like receptor 4 by flow cytometry analysis. Next, the secreted interleukin-6, MMP2, and MMP9 were quantified using ELISA assay. Last but not least, to assess the extent of apoptosis in triple-negative breast cancer cells, the co-cultured MDA-MB231 cells were assessed via annexin 5-FITC apoptosis assay. Moving on to the results and discussion part. As to achieve the first objective, macrophage cells were analyzed for their surface markers by flow cytometry analysis to investigate M1 macrophage differentiation upon Clinacanthus mutans treatment. Interestingly, Clinacanthus mutans treated macrophages showed an increase in expression of HLA-DR and toll-like receptor 4, cell surface marker for M1 macrophages. So this result indicates that Clinacanthus mutants could promote M1 phenotype differentiation in macrophages. Upon treatment of Clinacanthus nutans for 48 hours, the expression of matrix metalloproteinases MMP2, MMP9, as well as interleukin-6 were found to be significantly reduced. So this study showed that Clinacanthus nutans extracts may help in ameliorating the inflammation state between triple-negative breast cancer cells, MDA-MB 231, and also THP1 macrophages. In order to study the effect of M1 macrophages on triple-negative breast cancer cells, MD-MB 231 cells were co-cultured with 50% and 100% of Clinacanthus nutans treated macrophages, conditioned mediums. Results showed that the percentage of apoptotic MD-MB 231 cells was significantly increased upon treatment with conditioned medium, compared to the negative control, and also cells treated with Clinacanthus nutans only. So this result indicates that Clinacanthus nutans activated M1 macrophages effectively induced triple-negative breast cancer cell apoptosis. In conclusion, Clinacanthus nutans has the ability to reshape the tumor microenvironment by promoting the polarization of macrophages to an active M1 phenotype and suppressing the inflammatory cytokines, which in turn increases apoptosis of triple-negative breast cancer cells. Taken together, this study provides a rationale for utilizing Clinacanthus nutans extracts as a potential therapeutic target for anti-tumor immunity of triple-negative breast cancer. Together, we invest in hope for all women and our loved ones. Research brings us one step closer in achieving our goal to develop new effective targeted treatment to ease the burden of breast cancer patients. That's all from me. Thank you. Here is the list of references used throughout this presentation.